All right, let's dive in. Today, we're going to go way beyond your high school biology class to explore the fascinating and sometimes just plain weird reality of how our genes actually work. You probably remember those Punnett squares, right? Those neat little boxes with dominant and recessive alleles. Well, that was just the opening act. What if I told you the real story of genetics is so much more complex, more dynamic, and honestly, way more interesting? So, to kick things off, let's completely redefine what we even think dominance means. Turns out it's not always a simple case of one gene winning and the other one losing. Let's look at two concepts that really throw a wrench in that classic model: incomplete dominance and codominance. Now, they might sound kind of similar, but trust me, the results they produce are completely different.
First, you've got incomplete dominance. Honestly, the easiest way to think about this is like mixing paint. You have an allele for red flowers and an allele for white flowers. You don't get one or the other, you get a blend, a beautiful pink flower, right there in the middle. But then, on the flip side, you have codominance, and here there is no blending at all. Instead, both alleles get to show up to the party, and they're both fully expressed. The perfect example of this is our own ABO blood type. If you get an A allele and a B allele, you don't get some weird mix. Nope, you get AB blood type. Both are there, loud and proud.
Okay, so we've seen that even a single gene's expression can be pretty complicated. But things get even wilder when different genes start to interact with each other. So what really happens when one gene's job depends entirely on what another gene is doing? This is where genetics goes from a simple monologue to a full-blown conversation between genes. And let me tell you, the results can be completely unexpected.
And that brings us to this awesome concept called epistasis. It's basically when one gene can step in and completely silence or mask the expression of a totally different gene. It's like one gene has veto power over the other one, saying, nope, you don't get to be expressed today.
You see this all the time in the coat color of animals like Labradors. So you might have one gene that says, hey, the fur should be black or make it brown, but then a completely separate gene acts like a master on-off switch. If that switch is flipped to off, it doesn't matter what the first gene is screaming. No pigment gets made at all, and you end up with a yellow lab. And this is why this is such a big deal. It completely messes with those neat Mendelian ratios you memorized. Instead of that classic 9-3-3-1 you expect from a dihybrid cross, this interaction gives you this unique 9-3-4 ratio. It's like a secret code in the data that tells you epistasis is happening. Okay, now let's flip that idea on its head. What if, instead of multiple genes affecting one trait, you have one single gene that acts like a Swiss Army knife, affecting multiple, totally unrelated traits? That's pleiotropy. One gene might influence your height, your skin pigment, and your organ development all at once. It's super efficient, but it also explains why a single genetic mutation can cause such a wide range of symptoms. Okay, so far we've been talking about the genes themselves, the actual DNA sequences, but the story is so much bigger than that. In genetics, context is everything. The environment you're in, and even which parent you got a gene from, can change the entire story. Now, these two get mixed up all the time: penetrance and expressivity. Let's break it down. Penetrance is a simple yes or no question. If you have the gene, does the trait show up at all? But expressivity is more of a spectrum. If the trait shows up, how strong is it? Is it mild or is it severe? So it's the difference between an on-off switch and a dimmer switch. Here's a real mind-bender for you: genomic imprinting. This is the idea that for some of your genes, it literally matters if you inherited it from your mom or your dad. One of the copies gets stamped or silenced with an epigenetic mark, so only the other parent's copy is actually used. It's the exact same DNA sequence, but its origin story changes everything. So get this. What if you could have a trait that looks totally genetic, but isn't? That's what a phenocopy is. It's when some environmental factor, a chemical, a diet, a temperature, creates a physical trait that perfectly mimics a trait usually caused by a gene. It's a powerful reminder that we're not just our DNA. All right, for our last big topic, we're going to zoom way in on the chromosomes themselves. Because it turns out, in genetics, it's all about location, location, location. Where a gene physically sits makes a world of difference. Think about it. If genes are sitting right next to each other on the same chromosome, they're probably going to get inherited together, right? They're basically passengers on the same bus. We say these genes are linked, and they totally defy Mendel's law of independent assortment because, well, they're stuck together. But wait, that linkage can be broken.
During a process called crossing over, which happens when we make sperm and egg cells, our chromosomes can actually swap little pieces of themselves. This is huge. It shuffles the genetic deck, breaking up those linked groups and creating brand new combinations of traits. It's a massive source of genetic diversity.
And of course, we have to talk about the sex chromosomes, the X and the Y. This is what we call sex linkage. Because males only have one X chromosome, they don't have a backup copy for any of the genes on it. And that's exactly why X-linked recessive conditions like hemophilia or colorblindness are so much more common in men. All right, and to wrap this section up, let's quickly clear up two more ideas that sound alike but are totally different: sex-limited and sex-influenced traits. Now, these traits aren't necessarily caused by genes on the sex chromosomes, but their expression is controlled by whether you're male or female. It's pretty straightforward, really. Sex-limited means the trait only shows up in one sex. Think milk production in females or beard growth in males. But sex-influenced traits, they can show up in both sexes, but they act differently depending on the hormonal environment. The classic example is patterned baldness. The allele for it acts dominant in men, but recessive in women.
Whew. Okay, so after all that complexity, what's the big picture? What's the takeaway? Well, I think we need a whole new metaphor for genetics. It's not just a list of instructions, it's a symphony.
That's right. It's not some simple instruction manual where gene A makes trait B. It is a massive, complex, beautiful symphony. Every gene is an instrument, but how it sounds depends on all the other instruments playing with it, the conductor, which is the environment, and even the acoustics of the hall. It's this incredibly dynamic, interactive system.
So at the end of the day, our DNA isn't a fixed blueprint. It's more like a living, breathing script that's being interpreted every single second of our lives. And that leaves us with a really exciting and kind of profound question. As we keep learning the rules of this symphony, what part of that script are we going to learn how to rewrite next?